Hello and welcome to the Introduction to Pangenomics Cloud module. The Cloud module was created by the National Center for Genome Resources, or NCGR, located in Santa Fe, New Mexico. NCGR is part of the New Mexico INBRI, and the National Institute of General Medical Sciences, or NIGMS, supported this module. Pangenomes are collections of genomes from related individuals, often from a single species. Pangenomes allow you to capture, organize, and analyze genome variation across the species rather than relying on a single reference genome. That single reference genome usually only has a portion of the variation that's inherent to the species, and downstream analyses can be biased by relying on that reference. Pangenomes have become possible because of sequencing improvements. This graph shows changes in the cost to sequence a human genome since 2001. At first, the cost savings were tracking Moore's law, which essentially says the computer speed and capability doubles every 2 years. So, as Moore's law was bringing down the cost of computing, sequencing was following suit. However, the next generation sequencing revolution hit in 2007 or 2008, and you can see that the cost per human genome plummeted from about $100 million in 2001 to about $1,000 per human genome today. At the same time that the cost of sequencing was coming down, the throughput of sequencing increased rapidly. This enabled multiple high quality genome assemblies per species, allowing us to sample multiple individuals and the variation that they contain. In 2001, the first two human draft genomes came out. The public version that the government was funding came out in Nature in February of 2001. A day later, a private version was published in Science by Celera Genomics. The public human genome assembly was made up of a composite of different individuals. In this figure, you can see that most of the genome sequence, about 70%, came from a single individual. That individual had blended ancestry, so the assembly had ancestry from different regions of the world. The other 30% came from 19 individuals who mostly had European ancestry. On the right, you can see the different ethnicities and their proportions that ended up in the final version of the public human genome assembly. Along the way, we realized that there was more human variation to identify, and the genomes of additional individuals were sequenced as technologies improved. While the public genome was a mix of individuals, the Celera Genomics version was a single individual, Craig Venter. In 2008, James Watson's genome was published. James Watson was one of the co-discoverers of the structure of DNA. Both Craig Ventnor and James Watson are Northern Europeans, but most of the human variation resides in Africa. In 2008, an African genome was published. This was a Yoruban individual from Nigeria. In the same year, a genome assembly from a Han Chinese individual was published, and in 2009, we sequenced and assembled the genome of a Korean individual. At that point, we were starting to see some of the genetic variation among human individuals, and now thousands of high quality human genomes are publicly available, with many more on the way. This is a much more recent figure from the Human Genome Project looking at variation in the human leukocyte antigen HLA region. The colors represent different versions of haplotypes that go through this region from left to right, and the height of each bar is proportional to the number of people who have that haplotype. As you can see, there's a lot of variation in this region. This is one of the most diverse regions of the genome, but this is also a very important region because it is involved in immunity and human health and disease. Indeed, pangenomic analyses have implications for human health. Pangenomic analyses can help to identify the genetic origins of disease and characterize the evolution of cancers. They can also enable personalized medicine, which allows us to tailor treatment options to individuals and inform us about disease susceptibility. Pangenomic analyses can help identify stable vaccine and drug targets and have many more applications as well. In this module, you will learn about the field of pangenomics and use a small yeast dataset to walk through a pangenomic pipeline. You will build a pangenome graph using PGGB. You will then learn how to search the graph with BLAST and learn how to visualize and interact with the graph using Bandage. Then you'll learn how to use BG Toolkit to index the graph, map reads to the graph, and then call variants using those mapped reads. Let's begin.